Welcome back to Next Breakthrough, everyone. In this episode, I interviewed Dr. Anna Blakney, and she, similar to Dr. Kelly McNagny, who you met in the first episode, is a principal investigator at SBME at the University of British Columbia. So remember, SBME stands for the School of Biomedical Engineering. Dr. Blakney's lab is interested in RNA formulations and their delivery into the human body for vaccine and therapeutic purposes. I'm not sure if many of you are aware of this, but Dr. Blakney works closely with Dr. Peter Cullis, who was a pivotal part of the development of the lipid nanoparticle-based vaccines during the pandemic. Throughout this episode, we talked about this development and the continued improvement of self-amplifying RNA delivery Dr. Blakney and her team are working on. I'm beginning to realize how very exciting being the host of this podcast is. There is just so much learning you get in the span of one interview, and not only in terms of background knowledge, but innovative knowledge as well. If you guys share this feeling, please follow Next Breakthrough so you won't miss it. Fun fact, Dr. Blakney began a TikTok channel during the pandemic to answer the questions people had regarding vaccines. And without further ado, let's go ahead and meet Dr. Anna Blakney. I just wanted to start off with I know you run a TikTok channel in your spare time. Yeah, and I was wondering if maybe you could bring up your most commonly asked questions and then just give a really quick answer to them.
Sure. So yeah, I think the most common question that I get asked is about RNA vaccine safety and how they were developed so quickly during the pandemic. So I think, yeah, what a lot of people don't realize is just the years and years of development that went into them, and that actually the first clinical trial for an mRNA vaccine started in 2013. So that was well before the pandemic. So even though things were approved in a relatively short timeline, it doesn't mean that we were able to cut any corners. It's just that it's a very unique situation, right, in which all of the clinicians and companies and governments and regulators were all on the same page and focused on one disease, which isn't usually the case.
Okay, great. And then I think that's a great way to sort of ease into the more technical side of things, so your own personal research in your lab. So, could you start by giving us a simple breakdown of what gene delivery is? Because I know that's a really key component in vaccines, and why is it considered such a powerful tool in medicine?
Yeah, I think actually, I would consider gene delivery different than RNA vaccines. So I think it's actually really important to distinguish this. So with an RNA vaccine, you're trying to train your immune system to recognize a protein from a virus or some sort of pathogen, right? And so you actually need a very short exposure for that protein, which is why, you get one shot of an RNA vaccine and then another shot sometime later. And then maybe you need an update every year, depends on the vaccine. But with gene delivery, the exposure is different, right? So usually you're getting it because you're missing a protein or your body makes a protein incorrectly. And so you need it for a long time. And so you're likely going to get repeat injections with some frequency. So yeah, I think gene delivery, gene therapy, it's a bit different than a nucleic acid based vaccine.
Yeah. And I know you guys primarily work with the nucleic acid-based vaccines. Could you talk a little bit about maybe going back a few years, so right when COVID hit, maybe it was something that was emerging then? You talk about maybe what was happening then.
I mean, yeah, I don't think it was an emerging technology then. I think it's, I work really closely with Peter Cullis here at UBC, who's been working on this technology for 50 years. So it's been a long time coming, actually. I mean, in 1990 was the first time that somebody showed that if you inject mRNA into a mouse, that you could induce protein expression. So yeah, I think, it's for sure true that prior to the pandemic, we didn't have any approved mRNA vaccines. But there actually was an approved RNA drug with a type of RNA called small interfering RNA, and the drug is called Onpattro. And so, yeah, that's siRNA packaged in lipid nanoparticles, which is the same delivery system that we use for the mRNA vaccines. That was approved in 2018. So even as the pandemic started, patients were receiving monthly doses of that for like 2+ years, right? So I think it was the first time that a lot of the public had heard about mRNA vaccines, and that was, part of the reason for starting my TikTok channel. But it certainly wasn't a new technology.
Mhm. And I think because the public only knew about it, I think very recently you mentioned it also stirred up a lot of the concern for COVID-19 vaccines.
There's just nuances to that, right? So the way those clinical trials were performed, the main outcome that they're looking for is preventing severe disease and death. It's not just preventing any sort of like transmission or any sort of disease at all. And so that's what those vaccines are really good at doing, which is preventing severe disease and hospitalization and death. And in a pandemic, that's actually like the first priority, right? So I think there was a lot of questions about the vaccines and how they worked and just understanding that. And I think that's actually like not really new. Like I think there's people like Heidi Larson who have studied vaccine hesitancy, and I think would argue that it was very like highlighted in the media and apparent during the pandemic, but it's not like new ideas, certainly. I think it was just, all eyes were globally on these vaccines at that time. And so we all were thinking about it and talking about it more than we normally would.
Yeah, I think that's a great way to transition into the more cellular level of what is happening within those vaccines. And you mentioned that you work closely with Peter Cullis, and I think he was a really pivotal part of the development of the LMP-based vaccines. And I'm wondering if maybe you could talk about your role in your research within that time period as well, being a role in that development.
I was a postdoc at Imperial College in 2020 in the UK. And while I was there, I was part of a team that did the first phase 1 clinical trial of a self-amplifying RNA vaccine against COVID. So self-amplifying RNA is a slightly different type of mRNA. It's a type of RNA that's able to make copies of itself once it gets into a patient's cells. And so because of that, you can use a much lower dose, and hopefully it improves many things about a vaccine. One, the dose of RNA that you need, which will hopefully improve the side effects, but also the cost of manufacturing. And when you can minimize the dose of RNA, it just opens up a whole lot more possibilities.
Yeah. And since then, since your postdoc, I understand that you've opened a lab within UBC. What sort of research are you guys doing? How does it relate to what we've been previously talking about vaccines?
Yeah, so my lab works on self-amplifying RNA vaccines and therapies. I moved to UBC, yeah, you're right, in January of 2021. And so we still have lots of different vaccine projects that we're working on, but we're also very interested in RNA therapeutics. I think this is a big challenge in the field. There's still, I think, many ways that we could make vaccines better, and we're very interested in like understanding that and yeah, innovating in that space as well. But I think where this technology can really be impactful is like, a vaccine was kind of a low-hanging fruit for the technology, I would say. And so it's great that like the plane is off the ground, but now where do we go and where else can this make a difference? So that's what we're really keen to explore.
I know the main barrier that was identified on your lab website was delivering enough nucleic acid to the cells or tissues that you're working with. And then can you maybe help us visualize like what this means at the cellular level, and then why is it so difficult to get these molecules where we want them to go?
Yeah, I mean, there's a number of biological barriers that just like have evolved over millions of years to keep out foreign RNA, right? Your body views foreign RNA as a virus, and so as soon as it senses it, it sets off all these alarm bells, and yeah, then it starts fighting a virus or fighting your therapeutic or vaccine. Um, so I would say the main challenges in the field are that, so yeah, as I mentioned earlier, that drug Onpattro, that's a drug to treat there's like an aberrant protein that's produced in the liver. And it works really well in the liver because lipid nanoparticles pretty much go straight to the liver no matter what. So if you inject them intravenously in a human, within 15 minutes, all of the particles are in the liver pretty much. And so the big challenge in the field is getting them to go other places, right? So there's lots of different things you'd want to do. So just for example, like you might want to get RNA to the lungs to treat a cystic fibrosis patient, but it's very hard to deliver those particles to other tissues other than the liver right now. And even once it gets there, they're just not very efficient. So we estimate that 2 to 5% of the RNA actually gets into cells. And so you can just see it's like diminishing returns if most of it's going to the liver, and then only 2 to 5% of it actually being delivered into cells. And so those are the big challenges in the field right now.
Yeah. And then how is your team trying to target these challenges? So trying to get the RNA into the parts of the body that we want them to go. Is there something that we can tweak within their structure, their components that can make that happen?
Yeah, so we're trying to understand, like, I think there's a lot of kind of, I don't know, like endogenous ways your body does this and transports different particles around the body. So we try to learn from nature and then kind of adapt that to our synthetic systems. So I think there's a lot of like fundamental science that needs to happen as far as like understanding these mechanisms and what's governing that. And then we're also very interested in using like kind of more active targeting strategies, like attaching an antibody or a ligand to the LNP surface so that it primarily interacts with the certain cell type.
Hmm. And then actually, if we can zoom in onto attaching an antibody onto it, how would that like specifically improve the delivery into the body or where we want it to go?
Yeah, the way that these particles are taken up into cells right now is pretty like non-specific. So when you inject them into the blood, there's all these proteins in your blood, and through like hydrophobic or charge interactions, they just absorb to the surface of the particles. And then depending on what proteins have absorbed there, it dictates like what cells might take it up. And as I mentioned, like if you do this IV, now they primarily go to the liver. That's actually very well known. A certain protein in your blood absorbs to the LNP surface, and then that enables uptake into hepatocytes, which is a type of cell in your liver that then will express the RNA. So, yeah, that's kind of the like main understanding or like main way that yeah, we might want to tweak this and understand this better.
Mhm. Yeah, and speaking of problems that the delivery vehicle faces when it enters the body, can we maybe talk about the immune system's role? So I know the immune system is trained to recognize foreign components entering the body, and then how does it impact the treatment's effectiveness in terms of getting into the part of the body that we want it to go?
Um, yeah, so I think I don't know that your immune system is really like super active before it gets into cells. And so I think the biodistribution or like where it goes is more governed by like those proteins or like targeting strategies. But once it gets into cells, for sure your cells sense both the lipid nanoparticles and the RNA. And so yeah, if you remember a few years back, Drew Weissman and Katie Carrico won the Nobel Prize for the modified nucleotides for mRNA. And that's a way of kind of tricking your body into accepting the mRNA so that it doesn't recognize it as much at being foreign. So um, yeah, there's a lot of different strategies you can use to change the mRNA or the nanoparticles so that they're just detected less by your body.
Are there ways that you can maybe dive into that your lab is specifically trying to figure out for delivering these delivery vehicles into your body? The precise places that you want it to go. I know you talked about you mentioned antibodies attaching onto delivery vehicles. Is there like another maybe project that's going on that's another way to look at we can send these delivery vehicles into the body.
Yeah, that's mainly it that we're looking at for now.
Mhm. Okay. And then throughout all the research that you're doing within your lab, are there any big surprises or unexpected roadblocks that you've encountered?
Um, I mean, I think we find surprising things in the data all the time. Like that's kind of how science goes, right? Like you plan an experiment, you think the results are going to look a certain way, and then it looks totally different than you thought, and you have to just kind of follow that and move with that and understand why. Um, I think the biggest roadblocks for us is like, I guess, like a major bottleneck in our research is we would love to be able to just like print RNA, any RNA that we wanted, and have it be like super easy to make that. And it's pretty fast to like iterate on RNA designs, but there's still big bottlenecks in like designing and making new RNA. So in the fastest instant, you can probably do that in a month. But if you could just print that overnight, it would be such a game changer for us because we could just try so many more things and not have that limitation of time. So, yeah, I think if there's like kind of innovative and disruptive technologies that make that easier, then not just us, but the whole field will progress faster.
And then what specific diseases or conditions um, are you guys looking into when it comes with, because I know your lab focuses deeper understanding of delivery system interactions with the body. Is there a disease or condition that you're specifically looking at that requires these delivery vehicles?
Yeah, we're not really like we have projects for lots of different types of diseases, both infectious diseases, oncology. So we don't just have like one specific application that we're using this for.
Yeah. And I know your research also uses a wide range of models. I think that was mentioned in your lab website. So cell culture and as well as animals, I believe. And then how are you and your team working to develop better models? Or how do you use these models to actually predict formulations like efficacy within human bodies?
Yeah, I mean, I think it's tricky, right? All of our yeah, a lot of our work is in like preclinical mouse models, and like the reason for this is now there's, more and more systems of these like organ on a chip or organoid models. And the challenge is if you don't have like a complete immune system, you really don't know if your vaccine is going to work or not. And so I haven't really seen a compelling case of like being able to get around that at this point in time, although like obviously we would love to like reduce the number of animals that we use. Um, but the the major challenge with that is that mice have very different immune systems than humans do. And so often it's like really easy to make a vaccine that works well in mice, but it's not so easy to do in humans. And so we try to use like human cells and tissues as much as possible when we can in our research. Obviously, it's like not a complete immune system, and so there's limitations there, but if we use like primary human cells, then at least they have like intact innate immune system. And so that can give us some more insight and better answers for some of our work.
Yeah. So I think something that researchers are within your field are also looking at is the best way to find a model that actually translates well when it comes to the human body. I think that's maybe a topic that researchers are exploring in your field.
Yeah, I think yes and no. I mean, I think just the reality of it is that animals are always going to be different from humans. And so the question is really like how much proof of concept do you need to do in preclinical models before you just start a clinical trial with something?
Mhm. And can you maybe share a story from your research that had like a aha moment or unexpected breakthrough that completely changed your team's direction or changed something about the way that you continue this research?
I don't know that there's like a huge moment that sticks out, but I think like this happens in small ways all the time. Like there's just so many unexpected things that happen in your research, and like those observations are really important because yeah, you just can't, you never know how the science is going to go and how your experiment's going to go until you actually like try it out, right? And so I think what's really important is when you see those surprises, like following that, and it probably will alter your trajectory a bit to follow that. But that's often where like the most interesting questions and yeah, results are.
And if we could go back to the actual delivery of the mRNA, is there a reason why we use RNA and not DNA?
Like yes and no. I mean, actually, for a long time in the field, people like really preferred DNA over RNA. DNA, like it is much more stable than RNA, but the challenge, I would say, like the main challenge is that um for it to work, the DNA needs to get into the nucleus. And so it has to overcome like two biological membranes. And RNA only has to overcome one. And so because of that, yeah, it's just like typically easier to deliver an RNA. And then the other concern that people often have is the potential of DNA to integrate into your genome. And so RNA like doesn't really have that concern. But yeah, that I guess like that has been a worry in the past for plasmid DNA.
Because I know when you're delivering this vehicle into your body, is there a a challenge within that? Is there like toxicity concerns that can override the effectiveness that the vaccine can reach, the degree of effectiveness that the vaccine can reach to?
Yeah, I mean, with anything, if you like give somebody too much of it, you will see toxicity, right? Like even water. And so for any drug, like the most important thing is the safety and like the tolerability for the patient. So that's, I mean, for anything, you always want to use like the lowest efficacious dose possible. And so that's what we're always trying to improve is just to like make that as minimal as possible so that your side effects and tolerability are like very reasonable.
I think you mentioned this, but moving forward, is there a specific goal that you can foresee that your research or your lab is trying to reach currently? Because I know you guys made big breakthroughs during the COVID-19 pandemic. So I'm wondering for the future, what is something that you're looking forward to?
Yeah, so I mean, all of our research is really translational, right? And so we really like that's what I think motivates us as a lab is we really want to have a positive impact on human health. So what that means for us is really like getting things into clinical trials and then hopefully becoming a product one day.
And I know that artificial intelligence and machine learning are emerging into and are being translated into research. And I'm wondering how does it or how can it impact optimizing gene delivery in the future?
Hmm. So I think the challenge in our field right now is like if you look at the protein field and design, like they have amazing resources, right? With like AlphaFold and Rosetta and like all these tools that they have to like, design proteins. And they have that because they have big data sets to train those models on. And that just doesn't really exist for RNA right now. And so I think that's something that the field could really like benefit from is having more tools to understand how like the RNA sequence and structure affects its function.
And I know that you very recently navigated to UBC and created a lab. And I'm wondering for future researchers, maybe students right now that are looking into contributing to this field, specifically for delivery and vaccines, um, is there maybe a word of advice that you would give to them right now?
Yeah, I think just be curious and like try out lots of different things. I think that's like the most valuable part of being a student is you just have so many opportunities and it's a great time to figure out not just what you like, but also importantly what you don't like. And so I think just exploring as many things as possible is the best way to do that.
And last question, if you can maybe like look through everything that we talked about in this episode, so anything from it, and extract a a type of device that maybe could deal with some challenge in some area within your research, what do you think it would look like, and what would it be?
Oh, I don't know. Um, yeah, I'm not sure. Like maybe the closest thing would just be if you could like print RNA. Like that would be a big game changer for us.
I think through this podcast, one of the main things I'm trying to do is that give all these ideas to aspiring researchers and students that are focusing, that want to focus on this field, and maybe like create some discussion on something that we could build and develop for the future.
Awesome. Yeah, I think that's a great idea.
I think we can conclude our episode. Thank you so much, Anna, and I know it's a busy time for you. Thank you so much.
Yeah, it's great to meet you. Thanks so much. Bye.
Bye.